Okay, ready for this. We're diving into proteins today, but not just your average biology lesson. Think about it. What if we could design like custom-made proteins, to fight off diseases, create some crazy new materials? I'm talking engineering life itself. That's the power of these protein LLMs, and we're going to break down how it all works.
Yeah, it's a total game changer. For years, just figuring out how a single protein was structured, that was a huge undertaking. And now AI is stepping in. We're cracking the code of these amino acid sequences. They're like the building blocks, right? For all of nature's, incredible molecular machines. It's happening faster than ever before.
And we've got the inside scoop, excerpts from a brand new research survey on protein LLMs. Just came out this year, 2025. And get this, notes straight from the team that developed them. So we're talking cutting-edge stuff here. But the thing that really blows me away is how these models are going way beyond just understanding proteins. They're learning to design them, almost like writing code, but for biology.
Perfect analogy. Just like those language models, they learn from tons of text data. Protein LLMs do the same thing, but with protein sequences. They're basically absorbing the entire history of evolution, all encoded in those structures.
So hold on. Instead of writing poems or movie scripts, these models are composing proteins. I mean, how does a model even write something that, exists in the real world?
Well, it all starts with understanding the language, right? These models, they pick up those relationships between the amino acid sequences and the 3D structures they make. They're basically learning the grammar, the vocabulary of this whole biological code.
Okay, so it's like they're learning to read, like the blueprints of life. But how do you go from reading to writing? From just getting these proteins to, actually designing new ones?
That's where things get really wild. The early models, ones like UniRep and SSA, they used a kind of neural network called LSTMs, good at analyzing sequences, but then came the big breakthrough, transformer architectures. That's the same tech behind, those AI chatbots everyone's going crazy for.
Oh, right, like ChatGPT. So using that for proteins, that's what really kicked things into overdrive.
Exactly, like trading in your horse and buggy for a spaceship. Models like the ESM series, they're getting more powerful with every version, and it's because of this leap. Take ESM Fold, for example. Feed it a single string of amino acids, and boom, it predicts the protein's 3D shape, all those intricate folds, everything with crazy accuracy.
Whoa, wait. Just from a sequence of letters, it figures out those twists and turns of a protein, this whole 3D structure. That's like, I don't know, looking at a piece of flat paper and knowing exactly how it'll fold into a complex origami creation.
Exactly. And we're not just talking static structures. These models are taking on protein function too. We're not just seeing what it looks like anymore, we're figuring out what it does.
So we're going beyond just the form, straight to function. I mean, that opens up a whole world of possibilities, like targeting diseases or even designing proteins with like totally unique properties.
Absolutely. This area is still developing, of course, but some models are already predicting things like stability, solubility, even how a protein might interact with other molecules. That's huge for stuff like drug development.
Hughes is right. Instead of just testing tons of molecules randomly, you could use these models to design proteins with like very specific effects, like hitting a disease pathway but not touching healthy cells.
Exactly. And it's not just about predicting what's already there. We're starting to see protein generation models, ones that can create proteins that are totally new with the properties we want.
Hold up, we're not just tweaking existing proteins, we're talking about like from scratch design. How's that even possible?
Well, models like ProGen, they treat protein design like it's a sequence generation problem. You give ProGen your protein wish list, what you want it to do, and it tries to generate these amino acid sequences that fit your criteria.
Like a biological 3D printer. You put in the specs and out pops a custom-made protein? That's insane.
It's a revolution in how we approach protein engineering. And it all comes back to the fact that these models can learn from massive data sets. They're extracting the core principles of protein structure and function.
So the more data they have, the better they get at understanding that complex protein language, and then using that to create something totally new.
Exactly. And it's not just how much data, it's the types of proteins in those data sets. We need models that have seen the whole range, all the structures and functions nature's come up with.
So models that have basically learned from the entire history of protein evolution, from simple peptides to these like complex molecular machines. That's like giving them a crash course in the building blocks of life itself.
Exactly. And now they're using that knowledge to create proteins that go beyond anything nature's ever made.
This is all so impressive. But how do we know these models are actually working? I mean, are we sure they're not just spitting out random sequences?
Well, that's where all the testing comes in. We've got a whole bunch of ways to measure how accurate these models are, not just for predicting the structure, but for capturing that function too. And that's key, especially when you're talking about, medicine, where you've got to be sure it's reliable.
Right, you got to be confident that those results are trustworthy, especially if you're using them to design new drugs or therapies.
Absolutely. And the progress we've seen in how we evaluate these models is just as impressive as the models themselves. We're not just building cool tools, we're making sure they're accurate and dependable.
This is all so exciting, but we've barely scratched the surface. There's a whole universe of protein design and generation to explore. What else can these models do?
Well, we haven't even talked about multimodality, where these models can actually use different kinds of data, like text descriptions, structural information, even results from experiments, all to get a deeper understanding of these proteins.
So it's not just about that amino acid sequence anymore. We're combining all these different perspectives to get a more complete picture.
Exactly. It's like having different lenses to look at the same protein, each one showing you something unique. And this kind of multifaceted approach, it opens up even more possibilities, especially when you start thinking about combining protein LLMs with single cell data.
Ooh, single cell data. That's like getting a personal diary from every cell,
Right.
seeing exactly how genes and proteins are working in their natural environment.
Precisely. Now imagine the potential of combining all that detail with the predictive power of these LLMs. It could lead to personalized medicine like we've never seen before, understanding how proteins function at the individual cell level and tailoring treatments based on that.
Wow, so we're not just designing proteins in a vacuum anymore. We're starting to see how they work in the real world, in the complexity of a living cell.
Exactly. And that's where the real magic of protein LLMs lies. They're bridging the gap between these computational models and the messiness of real biology.
This is all so fascinating, but we're running out of time for this segment. Stay tuned because in part two, we're going to dive even deeper into how these models actually generate new proteins and explore all the mind-blowing possibilities they unlock.
We'll be right back. So welcome back to our deep dive into the world of protein LLMs. Before the break, we were talking about how these models aren't just reading the language of proteins anymore, they're starting to write it too, designing, brand new proteins with this incredible potential.
Yeah, it's like we've gone from deciphering those ancient scrolls to composing like whole new symphonies. We're not just tweaking what nature's given us, we're entering this era of like bespoke protein design. That's a huge shift.
Absolutely. And the survey paper we've been looking at, it highlights some really incredible examples of this. Remember how those early models, they focused mostly on just predicting the structure? Well, now we're seeing models that can actually generate these totally new amino acid sequences.
Wait, hold on a sec. We're talking about creating protein from scratch, like no natural template to go off of. How do these models even begin to tackle something that complex?
Well, a lot of these generative models, they use something called autoregressive modeling. Basically, they learn how to predict the next amino acid in a sequence based on the ones that came before it. It's kind of like proposing a melody, note by note, but using these rules of like protein harmony.
So it's like they're learning the grammar and syntax of proteins, right? Those underlying principles that govern their structure and function, and then using that knowledge to create something totally new.
Exactly. Take ProGen, for instance. This model treats protein engineering as like a sequence generation problem, similar to how language models generate text.
So instead of predicting the next word in a sentence, it's predicting the next amino acid in a protein sequence. But how does it know what properties those sequences will have? How does it ensure that, the new protein will actually do something useful?
Well, ProGen is trained on this massive data set of protein sequences and their annotations. And those annotations, they give you all this information about the protein, its function, where it's located in the cell, its evolutionary history, all kinds of things.
So it's like giving the model this whole textbook of protein knowledge, right? It learns from all the successes and failures of evolution and then uses that to design its own proteins.
Precisely. And because it has access to such a vast and diverse data set, it can actually generate proteins with properties that, we don't even see in nature.
So we're not just limited to what evolution's already come up with. We can explore all these totally new possibilities, proteins with like unique functions and characteristics.
That's the beauty of generative models. They open up this huge design space, letting us create proteins for really specific purposes, whether it's developing new drugs, designing like sustainable materials, or even tackling environmental challenges.
This is starting to sound like, science fiction. We're talking about creating proteins that can like break down plastic or, I don't know, capture carbon from the atmosphere. The possibilities seem kind of endless.
They are really exciting. And the survey, it highlights a bunch of other generative models too, each with their own strengths and ways of approaching the problem.
Okay, give us the rundown. What other models are out there and what makes them unique?
Well, there's ProtGPT-2, which focuses on generating these de novo protein sequences, with natural-looking amino acid compositions. It's kind of like making sure the protein language sounds fluent and believable to, like, a biological audience.
So it's not just about creating any random sequence, it's about making sure that new protein actually fits in seamlessly with the existing biological world.
Exactly. And then there's Progen 2, which builds on the success of the original Progen, but with an even bigger model and a much more extensive data set. It's like giving the model a supercharged brain and an even bigger library of protein knowledge to draw from.
So it's all about pushing the boundaries of what's possible, making these models more and more powerful and capable.
Precisely. And the researchers, they're always coming up with new and innovative approaches. There's UNC, for example, which uses a graph-based generative model. It can actually capture those super complex relationships between amino acids.
A graph-based model, that's interesting. How does that even work?
Imagine a network of nodes where each node represents an amino acid. And the connections between those nodes, they represent the interactions and dependencies between those amino acids.
So it's like a map of the protein's inner workings, right? The model learns how those amino acids are all connected and how they influence each other.
Exactly. And by understanding those relationships, it can generate these new sequences that still maintain, the essential structural and functional integrity of the protein.
This is all so fascinating, but with all this talk about creating these new proteins, I have to ask, how do we actually make them? Do we need to build like a protein 3D printer or something?
That's a great question. And it highlights the bridge between these computational models and, actually applying them in the real world.
Right, we can design all these incredible proteins on a computer, but how do we actually bring them to life, tangible and functional out there in the real world?
Well, there are a few different approaches. One is to use those existing techniques like recombinant DNA technology. That's where we insert the synthetic gene for that new protein into a host organism like bacteria or yeast.
So we're basically hijacking the cellular machinery of these organisms to produce our custom-designed proteins.
Exactly. And as these technologies advance, we're getting better and better at controlling that production process, making sure we get high yields and that the proteins are pure.
That's got to be crucial, especially if we're talking about things like drug development. You need to make sure those proteins are being produced reliably and consistently.
Absolutely. And there are other approaches on the horizon too. For example, cell-free protein synthesis systems, they let us actually produce proteins outside of living cells.
So no need for the bacteria or yeast. We can just like mix the ingredients in a test tube and watch the proteins assemble.
It's a really exciting area of research with the potential to completely revolutionize protein production.
It sounds like we're on the verge of this whole new era of biomanufacturing where we can create like custom designed proteins on demand, just like we print documents or manufacture plastics.
It's a truly transformative possibility, and it all comes back to the incredible power of these protein LLMs to decipher and then actually manipulate the language of life.
It's mind-blowing, honestly, but we're running a little short on time for this segment. Stay tuned, though, because in part 3, we're going to get into the real-world applications of these models. And we'll also explore some of those ethical considerations that come with this newfound power to, engineer life.
We'll be right back after a short break.
All right, welcome back, protein pioneers. We've been talking about how protein LLMs are not just reading the book of life, they're actually learning to write new chapters, designing proteins with, seriously mind-blowing potential.
And now we get to see those possibilities actually taking shape in the real world. These models are making a huge impact in so many fields already. From medicine to manufacturing, the applications are incredibly diverse.
Okay, let's get down to specifics. The survey paper mentioned a whole bunch of areas where these models are really excelling. What are some of the standouts for you?
One of the most promising areas has got to be drug discovery. I mean, traditionally, developing drugs has been like, trying to find a needle in a haystack. But protein LLMs, they're changing the game. They're enabling a much more rational and targeted approach.
So instead of just testing thousands of molecules hoping for the best, we can use these models to design drugs with that laser focus, right? Hitting a specific disease pathway without harming healthy cells.
Exactly. Imagine being able to design a protein that only binds to a cancer cell, delivering, a toxic payload while leaving those healthy cells totally untouched. That's the kind of precision medicine that protein LLMs are making possible.
That's incredible. And are there actual examples of this happening right now?
Absolutely. The survey mentions a study where they used protein LLMs to design these brand new antibodies against SARS-CoV-2. And these antibodies, they actually showed some really promising results in neutralizing the virus, so potentially leading to new treatment options.
Wow, that's a total game changer. And it's not just targeting viruses, right? These models could be used to develop treatments for all sorts of diseases, everything from cancer to Alzheimer's.
Exactly. And it's not even limited to designing the drugs themselves. Protein LLMs can also be used to optimize the delivery of existing drugs, making sure they hit the right target and minimize side effects.
So it's like having a GPS for medicine, guiding that drug right to its destination with pinpoint accuracy. That's huge for making treatments more effective and reducing those unwanted side effects.
Precisely. Researchers are using protein LLMs to design these protein-based nanoparticles. They act like tiny delivery trucks, carrying drugs directly to our cells. This targeted approach, it minimizes the damage to healthy tissues, which obviously improves outcomes for patients.
This is revolutionary stuff. And it's not just in medicine where these models are making a difference. What other fields are benefiting from this, like protein design revolution?
Biotechnology and biomaterial design, those are two big ones. Protein LLMs are being used to engineer enzymes with, enhanced catalytic activity, making them much more efficient at breaking down pollutants or producing valuable chemicals.
So we're not just treating diseases, we're using these models to create a more sustainable and eco-friendly world.
Exactly. And the applications go even further. Researchers are using protein LLMs to design new biocompatible materials, things like medical implants, creating artificial tissues that integrate seamlessly with the body.
It's like we're blurring the lines between biology and engineering, using the principles of life itself to come up with solutions to all sorts of challenges.
Precisely. And it all comes back to this incredible ability of these models to understand and manipulate the language of proteins. We're essentially learning to speak the language of life, using it to create solutions that, were pretty much unimaginable before.
It's an incredibly exciting time to be witnessing this merging of AI and biology. And as these models keep evolving and getting more sophisticated, who knows what incredible discoveries and innovations are waiting for us.
It truly is a transformative moment in scientific history. And as we wrap up our deep dive, I'm just in awe of the ingenuity of the researchers who are pushing these boundaries, What's possible?
It really is a testament to human curiosity and our constant drive to understand the world around us. And to our listeners, I leave you with this. What kind of protein would you design if you had the power?
The future of protein engineering is unfolding right before our eyes. And who knows, maybe you'll be the one to write the next groundbreaking chapter. So keep exploring, stay curious, and let's keep unlocking the secrets of the language of life together.
Until next time, keep diving deep.